So we have proteins in the blood that are outside the blood cell, and we have proteins that are in the cell itself. Inside the cell, we also have phosphate buffers. So this is most prominent intracellularly. But you'll also, it's a very strong filtrate buffer in the kidney. And we have carbonic acid, which is a very prominent extracellular buffer. And I'll draw some chemical reactions to show you how those work. In addition, we have systems, all right? So we have the respiratory system. By this we mean the lungs. And we have the urinary system. Okay, so we'll look at each of those. Respiratory system we've already talked about, but we'll review that. So here's an example of how an amino acid or a protein works as a buffer. Remember, to be a good buffer, it has to be able to either accept or give up a hydrogen ion depending on that situation. So amino acids have this acidic group. Amino acid has this component to it. And because it's an amino acid, it has a nitrogen. Okay? This is part of a protein. The rest of the amino acids would be tagged onto the end down here. Okay, so the R just means additional amino acids. Now, this would be a neutral pH for this particular amino acid. And each amino acid has a different pH at which it binds to and releases hydrogens. If we are in an acidic condition, okay, what would be the response of a good buffer in an acidic condition? It's going to take up hydrogen. It's going to remove hydrogen from the solution and bind it. And so that's what we see happening here. All right? So we don't add our extra hydrogen over here. We add it to the nitrogen. And we put an extra positive charge. So in an acidic condition, you would expect a buffer to have extra hydrogen ions. All right? In that same condition, we may or may not have that oxygen there. Well, put it that way, we will. Let me change that. We'll have that hydrogen there. All right? Now, in a basic condition, see if I can write this rather than have you redo it. So in a basic condition, there's not enough hydrogen ions in solution. So this one, we have too many hydrogen ions. Over here, we don't have enough. So we're certainly not going to take one and bind it to nitrogen. Instead, we take the hydrogen that was here and release it into solution. So we had a good buffer in a basic condition is going to release hydrogen ions, and that's what happened here. The hydrogen that was there was released. So we don't have an extra hydrogen here. We don't have enough hydrogens to bind a hydrogen there. What we do is we release the hydrogen that was on the acid end of our amino acid. Okay? So you should be able to recognize a protein or amino acid like that. If I showed you one that looked like this, you should recognize what condition it's in. It's given up a hydrogen, therefore it must be situation where the pH is too high, a basic condition. Here, we've added a hydrogen, so it must be an acidic condition where there's too many hydrogen ions available. This is driven by the amount of hydrogen ions, all right? Doesn't require enzymes, there aren't any hormones that drive this. It's concentration driven. Too many hydrogen ions, it looks like the diagram on the left. Not enough hydrogen ions, it looks like the diagram on the right.
Why is there no protein attached to the carbon?
Oh, there should be.
Oh, okay.
Okay? So that's how an amino acid or a protein works as a buffer. Okay? Follows our basic rule of accepting or giving off. So here our amino acid is a weak acid. Okay? Let's do the phosphate group first since I have that next. Can I erase this? Just the amino acid molecule. So this is showing a buffering of filtrate in the kidney, which was another place where we find phosphate. This is not inside a cell, okay, it's actually in the filtrate, but it's another place where phosphate is an important buffer. Okay, so this is one form of a phosphate buffer. Anybody in here work in a lab, cell lab or whatever? If you grow cell cultures, another form of this takes care of that extra charge there. And it's one of the most common ways of buffering cell culture media. If you're growing cancer cells or whatever, that's a very common buffer. So in this case, written like this, would that be in a basic condition or an acidic condition? Basic. The other form is that way. Or to complete the chemical formula, it would look like that. Okay?
That one's basic.
So are we adding or removing a hydrogen ion on the far left? We're taking a hydrogen out of solution and going from this to this. So we would only do that if we had too many hydrogen ions. Does that make sense? So this would be the result of an acidic condition. Because the extra hydrogen ions are being taken up out of solution. This would be in a basic condition because we're trying to lower the pH by adding free hydrogen ions. Okay? So when there's not enough hydrogen ions and it's in a basic condition, we release hydrogen from our buffer. When there's too many hydrogen ions, when the pH is too low, we bind hydrogen ions to our buffer. Okay? So here we're adding a hydrogen, here we've lost a hydrogen. Okay? So again, if the molecule has the extra hydrogen, it's buffering an acid. If the molecule doesn't have the extra hydrogen, it's buffering a basic condition. Whatever we need to do to adjust the pH. Right? And then I'll put on the board, you're already familiar with the carbonic acid, but I'll put it on the board anyway. So here's our weak acid. Okay, this is going to be in a acidic condition. And then as we move to a basic condition, we will release the hydrogen ion and try to lower the pH. So if somebody is in metabolic acidosis caused by eating a whole bottle of Tums, which are there to buffer the acid in your stomach, okay? So it's going to bind hydrogen ions. So if you ate a whole bottle of Tums, and I would suggest that you do, even if you're low in calcium, then are you going to have too many hydrogen ions in your solution, in your body, or not enough? Not enough. So you would be in alkalosis. So are your carbonic acid levels going to be, are you going to have a lot of carbonic acid, or are you going to have more bicarbonate and hydrogen? And in fact, what's going to happen to your respiratory rate? Is it going to go up or go down?
Up.
You're in metabolic alkalosis. You want to make more carbonic acid so you can make more hydrogen, so your respirations are going to decrease. They're going to become more shallow and less often so that you retain CO2 in your body. Okay? Is that a chemical buffering? What I just described, decreasing your respirations? That's the system, exactly right, John. That's using your lungs. Okay? So it's intimately related, but we have other sources of bicarbonate ions and hydrogen than just the lungs. So the chemical is certainly related to that, but it doesn't have to be. The respiratory system is changing the breathing. Do you actually breathe off hydrogens when you breathe off CO2? No, but you decrease the CO2 so you don't make the carbonic acid. All right, so let's look at the respiratory system then and what happens. So lungs, acidic condition, by lungs, we mean respiratory rate and depth. How fast you're breathing and how deep a breath you're taking. Okay? So your body is low pH. What would you expect your respiratory rate and depth to be? You have an acidic condition. You have too many hydrogen ions. So what change would you expect in your respiratory rate? Increased rate, increased depth. Okay?
So if you said that we don't breathe the hydrogen ions out, what do we do with them?
You don't do anything with the hydrogen ions except you don't use them to make carbonic acid. So you're getting rid of CO2. With less CO2, you're making less carbonic acid. The hydrogen is staying as water. Okay? So you're not, what you're doing is taking any hydrogen ions that might be there and adding them to hydroxyl groups to make water. So you're not breathing them out, but you're making more water than you are carbonic acid. Okay? If you're in a basic condition, let's say that you've been vomiting a lot, you're getting rid of the hydrochloric acid in your stomach contents, and the hydrogens aren't being reabsorbed back into your body, you're losing them in your emesis. So you're in alkalosis. Let's see, I'm doing the wrong thing. Let's say you have diarrhea, and you're losing the bicarbonate ions, okay? And, no, that was the right one, I'm sorry. You're vomiting, you're getting rid of the hydrochloric acid, so you're basic, you have too much bicarbonate from your stomach. Let me just kind of illustrate what's going on here, because we haven't done the digestive system yet. So, the stomach makes hydrochloric acid. That means your body absorbs those hydrogen ions as it moves through your digestive tract. In the small intestine, the pH, because the pH here is 1 to 2. In the small intestine, the pH has to be elevated to 8 to 9 from this 1 to 2. So to do so, the pancreas secretes into the small intestine bicarbs. So if we absorb those bicarbs from the small intestine, the two buffer each other, all right? If you're vomiting, you lose hydrogen, thus you have too much bicarb. Because you're losing the hydrogen and you don't have those to balance the amount of bicarb, okay? If you have diarrhea, you lose the bicarb. So you have too many hydrogen ions without the bicarb to balance them, okay? So vomiting is going to result in alkalosis. Diarrhea is going to result in acidosis, okay? So vomiting, not enough hydrogen ions because I'm throwing them up. Nice picture. So what's going to happen to my respiratory rate? I'm not getting hydrogen ions from my stomach secretions, so I have to get it by increasing my carbonic acid. So I'm going to decrease my respiratory rate and my depth, breathe more shallow. And that's how the respiratory system works as a buffer, all right? By changing the amount of CO2 and thus the amount of hydrogen from the carbonic acid. Okay, kind of a little bit of review of what we did with the respiratory system.
Oh man, that was just something we could set up one more time.
Sure.
I set my head wrapped around that.
Okay. So if you lose hydrogen ions, okay, in this case, because of throwing them up, not allowing them to absorb into the body, we're still making the bicarb. So we're absorbing the bicarb from the pancreas, but because we're throwing up the hydrochloric acid, this is not being balanced. Therefore, we have too many bicarbonate ions in our body. To help to balance that, the body will attempt to make more carbonic acid and release hydrogen ions from the carbonic acid. So the respiratory rate is going to decrease as well as the depth, therefore you're going to retain more CO2. That CO2 will form carbonic acid. The carbonic acid will dissociate into hydrogen ions and bicarbonate, and those hydrogen ions will buffer the bicarbonate that the body retained. Okay? All right, so that leaves us with the urinary system then. And we're going to deal with just one region of the urinary system, all right? Can I erase this on the board? Do you guys have it? All right. With the urinary system, we work directly with hydrogen ions. And we do so in the proximal convoluted tubule, the distal convoluted tubule, and the collecting ducts. Right? The distal convoluted tubule and the collecting ducts can go either way. They can secrete hydrogen or they can reabsorb hydrogen. All right? The proximal convoluted tubule can only secrete. We haven't talked about secretion yet, but that's what we're going to look at now for the proximal convoluted tubule. So we're going to go back and draw a diagram similar to what we did when we were talking about glucose movement. So we're in the proximal convoluted tubule. Over here will be peritubular capillary blood. And here will be filtrate. Okay. Now, we have a situation, reasons for which I can explain, similar to the respiratory system, in which our basal membrane, our basolateral membrane, doesn't allow hydrogen ions to cross the membrane. Remember that happened in our blood-brain barrier, in the brainstem? Hydrogen ions couldn't get out of the arterial blood. All right? On the apical end of our membrane, bicarbonate ions can't move into the cell from the filtrate. And yet, in an acidic condition, we have lots of hydrogen ions that didn't get filtered that we want to secrete into the blood. Remember, this is secretion. All right, and this is reabsorption. So in an acidic condition, we want to secrete extra hydrogen ions and move them out of the blood. That's going to lower the concentration of hydrogen ions and raise our pH. And then if we can take up bicarb, that will give us an additional buffer. We can buffer any hydrogen ions that we couldn't secrete. Okay? So this is an acidic condition. So in the blood-brain barrier, what did we move if we couldn't move hydrogen ions? CO2. So in the blood, hydrogen ions will combine with bicarbonate ions and form carbonic acid. This is taking place in our peritubular capillary blood. The carbonic acid is going to dissociate to form carbon dioxide and water. Now, carbon dioxide can diffuse into our cell. Okay? Within the cell, it combines with water and uses carbonic anhydrase, that enzyme that we had present in red blood cells. Okay, it's also present in our proximal convoluted tubule. Carbonic acid is formed again. Hydrogen ions can cross our apical membrane, and the bicarbonate ion returns to the blood. So now we've added hydrogen ions, but our bicarbonate levels stay the same because this bicarbonate that we used is returned to us. Okay? So we take hydrogen and bicarb, make carbonic acid, allow the carbon dioxide to diffuse in, make carbonic acid again, but this time the bicarb, which can pass the basolateral membrane, goes back into the blood, and we add additional hydrogen to our filtrate. Secretion of hydrogens. Okay. Not directly, we don't take hydrogen from the blood and add hydrogen to the filtrate, but we take carbon dioxide, and through the process of dissociation of carbonic acid, end up with more hydrogen in the filtrate and less hydrogen in the blood. Yes?
Sorry, what was the chemical you said?
Carbonic anhydrase?
Carbonic.
Okay, so but we don't stop here. We're obviously getting rid of hydrogen ions, okay? Which in an acidic condition is going to reduce the number of hydrogen ions and allow the pH of the blood to go up. We have these buffering systems in the blood, the phosphate buffer system, plus ammonium ions can be formed. And the purpose of this is just to allow us to add more hydrogens. Otherwise, we'd have a urine that, you know, like a pH of 1 or 2, and that would really burn. And we tried to go to the bathroom. So this way, when it's bound to the phosphate buffer or bound to our ammonium buffer, it's not going to change the pH, and we can add additional hydrogens. Okay? But we can make it work even better if we added more bicarb to the blood. Instead of getting rid of that bicarb in our urine, if we took the bicarb that got filtered and add it back to the blood, then we can buffer even more of these hydrogen ions. Does that make sense? Okay, so we have bicarb that was filtered. And now we're going to take it out and return it back to the blood. Moving from filtrate to blood is reabsorption. Okay? So we secrete the hydrogen ions and we reabsorb the bicarb. The bicarb won't cross the apical membrane. So once again, we form carbonic acid that dissociates to water and carbon dioxide, which diffuses into our cell, reforms using carbonic anhydrase into carbonic acid, which dissociates into hydrogen ions, which can't cross that membrane, so it comes back here. Therefore, there's no net loss of hydrogen, because we get it back again. And the bicarb now is added to the blood. Okay? So we lose our bicarb and get back the hydrogen ion that we used to make the carbonic acid. So now we've doubled the buffering effect of the urinary system. We're secreting hydrogen ions and we're adding additional buffers back to the blood by reabsorbing the bicarb. Okay? So, let's say that you have pneumonia. Pulmonary exudate is increasing the diffusion distance that air has to cross to get from the alveolus to the pulmonary capillary. And the carbon dioxide has to cross to get from the capillary to the alveolus. So by the time the blood leaves the lungs, not all of that CO2 has returned to normal. So our PCO2, which should have been 44, as it leaves the lungs, now is 50. Because we're building up CO2 in the lungs by not being able to breathe it off. With a higher PCO2, we're going to form more carbonic acid. What would be the normal response to higher amounts of carbonic acid and hydrogen ions? What would we normally do with the lungs? Breathe faster. Okay, but we've got pneumonia. That's why we've got respiratory acidosis to begin with, because breathing isn't allowing us to get rid of hydrogen ions fast enough. So those extra hydrogen ions are going to get secreted out of the urinary system. Okay? And we're going to reabsorb bicarbonate to help us buffer the ones that remain. Now, if we're in alkalosis, if we're at high altitude, how can high altitude lead to alkalosis? What are you doing at high altitude? Are you breathing shallow or deeper? Deeper to get oxygen. But then what you're breathing out is more CO2. So you might be getting in more oxygen, but you're also getting rid of more CO2 than you might be normally. We don't just breathe deeper and then breathe out shallowly. We have to move the same amount of air in and out. So we tend to get rid of CO2 at high altitude through hyperventilation or panic or having a baby, sorry guys, and breathing the wrong way, all right? And so we tend to get into respiratory alkalosis, all right? Are we going to do this in respiratory alkalosis? Would this help if we had respiratory alkalosis? No, it'd make things worse, because we'd be adding bicarb and we'd be getting rid of hydrogen. So remember, this is concentration driven. If we're in respiratory alkalosis, we're not going to have too many hydrogen ions. So this event doesn't take place. Not because of nervous system changes, not because of hormones, but because there's not enough hydrogen ions to drive it in that direction. So it doesn't take place in the proximal convoluted tubule. In the distal convoluted tubule, and collecting ducts, the opposite, in fact, may take place. We reabsorb the hydrogen and secrete the bicarb. Okay? Obviously, the basolateral membranes are flipped as far as what can be soluble across which membrane. So if we're going to reabsorb the hydrogens, we're going to have the We would see this and this on opposite sides of those cells to allow that to happen. Alrighty? So 3 sets of buffers. The chemical buffering taking place within the plasma, the extracellular fluid, or the cell, focusing primarily on proteins and the carbonic system. Within the cell, we have the phosphate buffers. Would protein function as a buffer? Would we see how come we don't see protein as one of the buffer systems here in the filtrate? Protein doesn't cross, exactly right. Protein, unless we have a damaged glomerular filtration membrane, protein doesn't cross. So protein would work as a buffer in the peritubular capillary bed, but not in the filtrate. Okay? All right, so let's take a break and come back at 2, and then we'll do the Henderson-Hasselbalch equation and figure out how you can determine whether somebody is a respiratory or metabolic acidosis or alkalosis directly related to the exercise worksheet. Okay?